Hello everybody, I'm here to talk to you about our project, Microbial Source Tracking Water Pollutants, and how it reveals why animals are not to blame for the pollution. And this work was done by me, Ian Ball, who was unable to make it today, and Dr. Joshua. And let's get into it. So water is one of the biggest reasons we're all here, and it is a very vital source of recreation, and also we drink it as well. So the things that pollute it are very important to study and focus on to improve community health. And so a little bit of background about this project. We studied the East Fork Whitewater River. Here in eastern Indiana, it runs through Richmond from the reservoir and south of town into Brookville. And again, it is important for recreational uses and ecological purposes. And so this waterway has actually been placed on the Clean Water Act Section 303D list. And that basically just says that our water is above the threshold level of allowed E. coli levels, and it's unhealthy because of this. And so we really wanted to study and figure out where that's coming from, which is our research question: where is the fecal contamination coming from? And that's what we focused on in the study. And so most warm-blooded animals have E. coli in their gut, and we know this. Most of us have heard of E. coli before, specifically humans, geese, cow, pig. This is a specific bacteria that is used to determine fecal pollution in waterways, but we also have a bacteria called this Bacteroideus, and this is also in the warm-blooded animal gut as well, so it can be also related to fecal contamination as well. And it has a special region in its ribosome that we're able to differentiate between hosts. It's called the hypervariable region, and so this can be kind of used as a target to figure out which host it came from. And so we'll start with how we went ahead and analyzed and collected our samples. We'd go out to the water. We would collect a 1 liter sample as well as some water parameters. Then we bring it back to the lab, and we had a vacuum filtration set up here that we would filter the water through. We would culture some of these filters, and then we also extracted DNA, which you can see Ian doing here. And we would also do a qPCR analysis. So for sampling, we have 6 sites in Richmond that we have strategically placed, one at the reservoir where we see a lot of recreational use, and as well the Indiana American Water Treatment Facility is right here. It takes from the outlet of the reservoir for our drinking water, and they clean it up. Site 2 is just after that, and then Site 3 is where these 2 streams converge. So if we find that Site 3 were to have high levels, we could go back up this stream if we didn't see them here at Site 2. Same thing for Site 4. There's a residential area in here, and we could go back up this stream if we find there's no pollution this way. So it really allows us to track where it's coming from. Site 5 is downstream of more residential area, but it's right before the water treatment facility for sanitation or sewage. And so we wanted to get a baseline of what the water looked like before we came into the treatment plant, and then Site 6 is directly afterwards. So we're able to see kind of increases or decreases after that. And some of the water parameters are listed here: pH, DO, RP, and we collected these to see if there was any correlation with what we found to those parameters. And we collected this data for 1 year, and we're still collecting it. So for filtering, this is the DNA extraction. We did a little bit more water than we did for the culture method, but basically we used a specific filter that was able to filter the water and leave everything else from the water on the filter. And so we were able to put that on special auger plates that either showed us fecal coliforms or just total coliform numbers. And then again, also we did the DNA extraction with these filters. And the qPCR analysis allows us to figure out a concentration of the bacteria that we found in the sample. And so we use primers, which are short, specific, basically they target DNA specifically, and we have one specific for each different type of organism. We did human, Canadian geese, cattle, and pig. And we were able to basically load our samples in this plate here, run it through a machine, and within 2 to 4 hours we would have results, which this is important because previously people are using the culture method, which takes at least 24 hours to return results. So this is a much quicker method of finding results, and you're able to figure out specifically what host organism it's coming from. And so here's some of the results that we had for our culture method. Because we still did do that, we wanted to make sure that our findings were consistent with the qPCR that we were doing, and we found that we still had elevated levels of total coliform counts and fecal coliform counts. And I wrote down the rankings. We actually ranked the sites based off of geometric mean, and so we found that based off geometric mean for total coliforms, site 6 had the highest, and basically it worked its way backwards: 6, 5, 4, 3, 2, 1, highest to lowest. And then for fecal coliforms, it went site 6, then site 3, site 4, 5, 1, and 2. And so that was an interesting finding, and we're still doing a little bit more analysis on as to why that is. But we expected to see a little bit of compounding to happen as the water runs. We would see more bacteria building up. And then here is the qPCR results for human. And so we found in 99% of our samples that we ran, we detected human fecal pollution. So that is not a good thing. I know here it says sites 4 and 6 had the highest levels, but we've actually done further analysis, and I wrote these numbers down too. Based off of geometric mean, the ranking went 5, then 3, then 6, 4, 2, 1. Which also was an interesting pattern. Yeah, so there's that. And next we did the GFD, the Canadian geese, and I have this circled here. Well, we found 60% of our samples were detected positive for Canadian geese. And I also have this circled here because we plotted these against precipitation and temperature. And so this dotted line here represents precipitation. And we had a heavy rainfall event right before the sampling date. And we can see that all of our samples were elevated for bacteria, for the Canadian geese here. And this was kind of consistent with the rest of our results as well, but this specifically showed it most elevated. And so we found that precipitation might have a large effect as well on what we're finding. And then the third lowest contamination I guess we found was the cow, and we found that this was detected in 25% of our samples. And another interesting finding is that we actually only find it about late fall into late spring. And so we've done a little bit of analysis with this as well as pig, which I'll go over next, but we're starting to think that this might be due to manure being spread and then runoff throughout the winter on the fields. And then pig marker, 16.7% of our samples, this was the lowest contributor to our findings. And again, it follows that seasonal pattern of late fall into kind of late spring. And so our conclusions are that human-associated marker was detected the most, and so humans are going to be like the highest contributor to the fecal pollution in our waterway. And then next up will be the Canadian geese, which we kind of expected. We see a lot of those along the reservoir and all throughout the waterway. Yeah, they're all over. And so multiple organisms contribute to this, but it really does appear that we are the biggest problem, unfortunately. And so the implications of this work is that we can use this to be able to kind of focus in on watershed management in the future. Like if we know that humans are causing the problem, we can't really pass the buck anymore. And so we'll have to figure out what exactly is causing that human pollution to seep into our water. And then again, the qPCR based method allows us to specifically identify that pollution and in a much faster method. And then future research, we're just going to try to continue sampling, investigate where that human bacterial contamination is entering, and try to analyze water samples from upstream, those other directions I had pointed out earlier, to see if those are contributing. And then this is a little bit of community engagement that we've kind of fallen into while out sampling. We ran into an administrator from the Richmond Sanitary District, and they're having like a wider 2-year watershed study, and they invited us to run these microbial source tracking analysis on their samples as well. And so this is really a greater impact on our community, and it has been a way to help also fund future students to continue learning this work and kind of help our community out. And I'd like to acknowledge my principal investigator, Dr. Jia, and this study was supported by the senior research grants of the Indiana Academy of Science, so they've been very helpful in our study as well. And then the Faculty Research and Creative Activity Support Fund from IU East. Thank you. Yes.
Hunter, that was just great. And,
thank you.
And good for you, Dr. Zhao, for really getting this going to help our community. I'm glad to know that you are working with the sanitary district and with Mr. Smoker, Pat Smoker. Is he aware? It just seems like this has a lot to do with our sanitation and overflow, right? I mean, we're supposed to be keeping our poop, covered, buried, and out of our rivers. So what's going wrong? And are you working with do the right people who can change that now know about your study?
Yeah, so we have had kind of group meetings with Earlham as well as Pat, and Leanne is another contact we have out there. And they do know that there are these elevated levels. Pat has told us that actually the overflow system that they have in place is supposed to be able to handle the capacity even during the worst of storms in Richmond. And so other factors may be contributing, like leaking septic systems or I can't speak to people dumping directly into the water, but these things can happen, and so they do know that this is a problem, but since we're able to finally track down that source and point fingers eventually, it'll be more helpful in mitigating it.
Glad to clear the animals' names, though.
Any other questions for Hunter? You can
Yes.
go ahead.
A lot of that was Greek to me, and I'm just going to admit that right now. But one of the things I was wondering about is, did you see any patterns in time of year, in the sense of, like, I'm just thinking, like, times when we might see greater concentrations, particularly from animals?
Well, so again, we had the kind of the seasonal, pig and cow. So we see more positive samples kind of in the late fall and late spring, because I believe they're spreading this manure on these fields and it's kind of running off into our waterways. And so for animals specifically, I think that's probably when we'll see pig and cow increase. The goose was kind of always there, it seemed like, for the most part. So I think the precipitation is going to be the bigger factor rather than the seasonal part. Yes. Yes.
Have you looked back at data from the 90s, 80s, 70s to see if that level has increased or if that level has changed? And then I wonder if there's a correlation between the change in those numbers and the American diet. And the foods that we consume and things that may lead to an increase in those numbers.
Yeah, we have not done any we haven't done any retro analysis or anything on any records that were kept, but that is an interesting idea, and maybe that could help establish our study a little further in the future. So thank you for bringing that up. Yes.
One of the other things I was thinking of is, I guess, not that there's a huge part of this in Richmond, but there is still any of the locations, I guess, that have some of the higher, are they associated with any areas that possibly there is homelessness and, people being by these water systems because that is where they're sitting to,
Yeah,
There's that direct.
Yeah. I actually don't. I don't know. I haven't seen many homeless, like, I'm sure there are homeless people, but I don't know what areas those would be specifically, so I'm not sure. But that's a good point to maybe look at further. That's true.
Do the levels by humans differ by season? Do they change from season to season?
I think that the levels were always there, so it was a little bit harder to see the seasonal analysis in humans, if you know what, do you know what I mean? Like, it was always there. We did see, we had a very large spike, like, in the very beginning of our study, so that's kind of midsummer time, but I can get back with you on that in the future. Awesome.
Thank you for all your questions. Thank you, Hunter. Looks
Thank
like
you.